All right, so last week we ended talking about the types of antibiotics there are and their major targets within bacteria. So this week we'll wrap up our bacteria section by talking about resistance to antimicrobial drugs, and then we'll talk about a concept called the parvome. And so we've talked a little bit about the biosynthesis of these types of antibiotics within the bacterial communities, looking at our PKS, our macrolide antibiotics, and their production through the polyketide synthases, or the PKS domains from last week. And one of the key things that I wanted you guys to take away from those lectures was that the polyketide synthases work in a very similar way to protein synthesis in the ribosome. You've got these very large protein complexes that come together and they start building these very large natural products by the addition of small units that they link together. Just like you have amino acids that come together to make up your protein, you've got the ketide extended or 2 carbon units that come together to form your polyketides. There's also non-ribosomally encoded peptides as well, and we saw a few of these guys having antibiotic activity as well. And so these guys are not produced on the ribosome, but they also have a very large protein complex that's involved in their production that will link in each amino acid along the chain. Only these amino acids don't have to stay in a linear arrangement like they do in proteins. They can be cyclized and they can be highly modified amino acids. And so this gives structural diversity to the classes of natural products that we'll be seeing throughout the term. So for all the natural products that have been isolated from natural sources, including bacteria, including animal sources, and including plant sources, each of these organisms has to have biosynthetic machinery, or the proteins available, to actually produce these natural products. And so the biosynthetic machinery that enables these different organisms to make natural products is actually called the parvome. And so we will spend some time talking about the parvome, especially in our consideration of antibiotic production, and where we're going to actually find new antibiotics to help us start fighting these resistant microorganisms. So drug resistance really limits the use of all known antimicrobials. Penicillin G was the first antibiotic introduced, and when it was introduced, only about 3% of the bacteria were resistant to it. Now over 90% are resistant. And we're seeing similar things for our other antibiotics that are being used clinically. So this is a very large problem. So how do the bacteria become resistant, and what can we start doing about it to try to eliminate the resistance or combat the resistance? So bacteria can have spontaneous mutations. This happens as the cells replicate, and so it's basically an evolutionary process. So natural selection is going to select mutants that have higher propensity to survive in the face of the selection process, which in this case is going to be the antibiotic. And then bacteria, as we said at the very introduction of the bacterial section, have this propensity to have horizontal gene transfer. So they can transfer plasmids and they can transfer chromosomal DNA from one bacteria to another bacteria based on the pili that are formed by a bacterial species. And this horizontal gene transfer can take place within a species, or it can take place between species. And so this really allows antibiotic resistance to spread very rapidly and widely among many different bacterial species, because they can obtain and utilize the resistance genes from one species through this horizontal gene transfer process. So when thinking about natural selection and the evolution of antibiotic resistant bacteria, what you can see is that if you have a dish of bacteria that are growing and you add antibiotic, you'll see that the antibiotic will start killing most of the bacteria. However, if there are few bacteria that have a mutation in them, so that would be these little red bacteria that are sitting here, if these guys show resistance to the bacteria, they will actually start multiplying and growing out, because they will have the selective advantage to being able to grow in the presence of the antibiotic. And so you get a resistant bacterial culture. So you'll often hear doctors tell their patients to take and complete their entire antibiotic course when they're on antibiotics. And this is because at the first go, you likely don't have any resistant bacteria within the population. However, if you don't maintain a minimal inhibitory concentration of the antibiotic within your system, you may be enabling mutations to occur within the bacterial population that isn't being hit hard enough with the antibiotics to kill them straight off, so that they become more resistant to that antibiotic. And this can cause resistant bacteria to actually outgrow from an infection. And this can be very dangerous because now the bacterial population is not going to be responsive to that antibiotic. This is not the only way, though, that bacteria become resistant to antibiotics. The presence of antibiotics within the environment is also a huge problem. So if antibiotics get into our environment, especially within our waterways, so if they're contaminating like rivers and streams, they can rapidly spread antibiotic resistance to other bacterial strains because the bacteria actually get exposed then to very low doses of antibiotic. It's in a very diluted way when it gets into the streams, and it's going to cause those bacteria to actually be able to develop immunity and resistance to these antibiotics. And so our agricultural systems actually contribute a lot of antibiotic content into the environment. And it's because they put antibiotics in the feed of many commercially grown agricultural species, including cows, pigs, and chickens primarily. And that's because we're overloading our farms with these animals. We're trying to grow too many animals in too small a space, and they suffer more from bacterial infections and disease. And so to help combat that, the farmers are putting much more antibiotic concentrations into their food as a preventative measure. So they're not using them to treat disease, they're using them as a preventative. And this is increasing the load then of the antibiotic structures that get into the environment and can lead into antibiotic resistant strains of bacteria. What types of biochemical mechanisms then are these bacteria predominantly utilizing to gain resistance? First, they can have drug inactivating enzymes, and so we've already seen this with our beta-lactamases. So these guys are going to actually disable or inactivate the beta-lactam antibiotics, and we saw that these enzymes are very good at just breaking open the lactam ring structure, and then the molecule or the natural product is no longer active. So penicillinase is a beta-lactamase that would break down this structure, and it is a very good example for this type of mechanism. Some bacteria will actually alter the target molecule. A lot of the bacterial agents that we talked about are actually targeted at the ribosomes of the bacterial species, and so resistance to these compounds has largely been shown to alteration of the actual ribosome structure. So there might be mutations in the genes that encode the ribosomal RNA or the small proteins that also make up the ribosome. And so if these get altered, they can get altered in such a way that they no longer will bind to the antibiotic compound. And so now the bacteria has developed resistance to that antibiotic because the ribosomes are still functional, but they'll no longer bind to the antibiotic compound. So another way that bacteria can become resistant to a drug is by decreasing the uptake of the drug. So in this mechanism, you would have increased elimination. And so there would be an increase or alteration in porin proteins. And so these are usually ABC transporters that we talked about in, I believe, week 2 or 3 when we were talking about how drug molecules can get into cells. Well, these ABC transporters can actually eject molecules from the cell as well. And so if they have alterations in these proteins so that they specifically will recognize the antibiotic structure, the minute that the drug actually comes into the cell, it will be grabbed by the ABC transporter and it will be kicked right back out. And so by this mechanism, the drug has no time to enter the cell, bind to the ribosome, and inhibit the production of proteins within the bacteria. Instead, it just enters and gets kicked back out through the ABC transporter. So this would actually be an increased elimination of the drug. Decreased uptake would be blocking of the drug from entering into the cells. So you'd have decreased uptake shown actually right here. So you may have a porin protein that prevents entry into the cell. So the porin is actually that used to mediate passage of the drug into the cell has been altered, and now that drug molecule can no longer get in. So all of these different ways are biochemical means that the bacteria can evolve to actually become resistant to the drug compounds. So how do we slow the emergence and spread of antimicrobial resistance? There's a responsibility of physicians. They have to be able to identify microbes and start prescribing suitable antimicrobials to their patients instead of just over-prescribing antibiotics. So oftentimes, your physician might say, you're suffering from a viral infection, antibiotics aren't going to help, and they stop prescribing the antibiotics. Some responsibilities of our patients, they need to carefully follow the instructions that are given by their physicians to actually complete doses of antibiotics when they are given. And then responsibilities of the agricultural industry is also very important. This is probably a huge area where we could really curtail the use of antibiotics so that you have bigger elimination of the introduction of antibiotics into the environment. We need to also educate the public so that they understand the appropriateness and limitations of the antibiotics. Again, antibiotics are not effective against viruses. So if you have a viral infection and you have like the common cold, antibiotics aren't going to do anything to help you eradicate that cold any quicker. And then global impacts. If you have an organism that is resistant, it can quickly travel to another country. And so how do you prevent this? In some countries, you have antibiotics available on a non-prescription basis as well. This can also be problematic. So global impacts. Anymore, our world is really becoming a global population. Travel is so easy around the world anymore that many people come into contact with each other that never would have had access to each other. And so an organism that is resistant can quickly travel from one country now to another just via airplane routes. This has been a huge concern with things like drug-resistant strains of tuberculosis. The CDC has actually banned people in the United States that have drug-resistant forms of tuberculosis from flying. They won't let them actually travel on the airways because they don't want them spreading this dangerous bacteria that they're infected with. In some countries as well, like Mexico, antibiotics are available on a non-prescription basis. So you could just walk into any pharmacy and ask for Cipro, for example, and they would give you Cipro. So this actually isn't the best way to administer antibiotics. So as you've seen in our previous slides, that the use of antibiotics is highly selective to the type of organism that you might be infected with. And the general public generally doesn't have a good enough education based around the types of bacteria that they might be infected with to choose the correct antibiotic that will best serve their needs. And so this actually can be a big problem. And then our antibiotics that are fed to animals can select for drug-resistant organisms. Again, this is probably the number one problem that we have for pushing antibiotic resistance in bacterial populations. It actually puts a huge amount of antibiotic load into the natural environment in our waterways just through runoff from the feed and feces from our domesticated animals. So what's one way that we can try to combat some of this resistance? You can use synergism. So oftentimes, more than one antibiotic given in combination can overcome the resistance of a specific strain. And synergism occurs when you actually have a response that is greater than the effect of either substance given alone. And so an example of this, if you give penicillin and streptomycin in the treatment of something like bacterial endocarditis, so this is a bacterial infection of the heart lining. It's also very hard to treat, but by using these two antibiotics, you get a synergy between them. And the damage to the bacterial cell walls by the penicillin actually makes it much easier for the streptomycin to enter the cells. You get a much higher concentration of the streptomycin inside the cells, and this increases your rate of cell killing. But you have to be careful. Some combinations of drugs can be antagonistic. So if you give penicillin and tetracycline together, it's often actually less effective than when you have either drug used alone. So this should be either instead of wither. By stopping the growth of the bacteriostatic drug tetracycline actually interferes with the action of the penicillin. So the penicillin requires actively growing bacteria so that it can actually inhibit the cell wall biosynthesis. If the cell wall is already hooked together and the bacteria are just sitting there in a dormant state, then the penicillin is not going to be able to mediate a toxic effect to the bacteria. So this would be an example of where you would have an antagonistic combination of drugs that would not be helpful. So you wouldn't want to use tetracycline and penicillin together. So when should combinations of antimicrobial drugs be used? They should be used to prevent or minimize the emergence of resistant strains, to take advantage of the synergistic effects, and to help try to lessen the toxicity of individual drugs. So sometimes when some of the antibiotics have very high toxicity, if you use them in combinations, you can actually reduce the dose that you need to be effective to have a bacterial kill, and you reduce the overall toxicity to the patient with that antibiotic because you can use less. So this would be another useful way to use combinations of antibiotics in treatment. And then new approaches to antibiotic therapy are needed. And so in our next lecture section, we will take a closer look at some of these ways that scientists are trying to exploit antibiotic production from other organisms and to try to get at new antimicrobial compounds. So one way of trying to determine drug susceptibility of specific bacteria, and this would be a way that a doctor could actually test a microbial strain that would be isolated from a patient. The bacteria is going to be spread on the surface of an agar plate, and then 12 discs, each containing a different antimicrobial drug, will be placed on the agar plate. The plate will be incubated to allow the bacteria to grow, and the drugs that are impregnated onto these 12 discs will diffuse outward and they'll kill the susceptible bacteria. This is called a zone of inhibition. So the larger the zone of inhibition, the better the antibiotic is working to kill the bacteria. So you can compare this size of zone to a chart based on normal antibiotic activity. So you can look for a drug-resistant strain, and then you would know exactly what to treat your patient with. So this would be an example of such a treatment. So each of these little discs has a different antibiotic in there, and you can see when the bacteria is streaked here that the antibiotics that are effective at killing the microbe will have a larger zone of inhibition. So they will inhibit the growth of the bacteria within this region. So you can see these two antibiotics have very little effect on the bacteria, whereas these two antibiotics have a big effect. So if I was treating a patient clinically and had done this screen on the bacterial strain they're infected with, I would want to choose one of these antibiotics to be my primary treatment option. So this can also be a way that new drug screening protocols can be tested as well. If you have pure compounds that you want to test, you could use a control sample where you might have an antibiotic of known concentration and known toxicity, and then you could compare this to a drug of interest that you have just isolated. So finally, what are some clinical considerations for treatment of bacterial infections? So when thinking about antibiotics, you have to think about their availability within the region, their expense. So within the United States, we actually have a lot of availability to a lot of different types of antibiotics. But if we were in a third world country, we'd have much less availability to different antibiotics, and expense would be much more of an issue. You also have to worry about the stability of the chemical so that you can travel and take it to different places within the world and still maintain its activity. It has to be relatively non-toxic and non-allergenic to be a very popular antibiotic. And it has to maintain selective toxicity against a wide range of pathogens without being toxic to the host. So we've talked about a lot of different types of antibiotic compounds, and really we've been on this side of the spectrum here. So the antimicrobial drugs listed here, you can see their range of activity. And so these are called their spectrum of action. So we talked about some narrow spectrum antibiotics, like the penicillins, that are mostly going to be effective against gram-positive bacteria, and the polymyxins that are mostly going to be effective against gram-negative bacteria. And then there's also some broad-spectrum antibiotics that are reactive against a large range of microbes, such as the tetracyclines and the erythromycins. And then we haven't talked about this area yet, but we will be going here towards the end of the week, talking more about compounds that are used to combat eukaryotic parasites. And one that we'll focus on are going to be the malaria parasite. And then we'll talk about some viral treatments as well. So I think we've already mentioned this thought question. Why is the use of a broad-spectrum antibiotic not always desirable? Well, if we think about the role of our normal microbiota, they play important roles in things like digestion and food absorption especially. And so we often have side effects from our antibiotics because we're killing off our natural microbiota or our natural microflora. So broad-spectrum antibiotics aren't always the most desirable. If you know what type of infection you have, you would much rather use a specific antibiotic to treat that infection. So safety and side effects, these are also important considerations that clinicians and physicians need to make and be aware of. So for example, we mentioned the polymyxins and the aminoglycosides having toxic effects on the kidney and also on the ear. So these can be fatal effects as well. And you just see some of the side effects here from different compounds, including the azoles and the tetracyclines, causing discoloration of teeth in this case. And then of course allergies, and then complications caused by disruption of normal microbiota. They can allow other opportunistic infections to occur. So things like Candida albicans can infect the vagina or the mouth. And this often increases when you use broad-spectrum antibiotics. So these are considered superinfections due to their uncontrolled growth. And one thing that we should mention about antibiotic-resistant organisms, in the absence of the antibiotics, resistant cells typically are less efficient in growth compared to normal cells. And this is because there's an expense to making resistance components within the cells. They have higher metabolic requirements because oftentimes they have to make new proteins, or they have to alter their protein structures, and these altered protein structures might not be as biologically active as the wild type components. So there's a cost to becoming antibiotic resistant. So antibiotic resistant populations will usually only come into being if antibiotics are present, so that the normal cells die and open up a niche for these resistant populations to take their place. So some of our thought questions that you should now be able to answer are the different mechanisms that bacteria can use to obtain antibiotic resistance. So you should know at least 4. And another thought question, why do you think resistant strains of bacteria develop more often in hospitals and nursing homes? So what you probably surmised is that more infected people come to these places. And so if you have a higher propensity of seeing infective strains, then you're going to have more resistant strains that are going to develop. Hospitals and nursing homes are also the sites of treatment, and so you're going to have higher levels of antibiotic use within both of these facilities, and that will also promote the development of antibiotic resistance within the bacteria. And then we'll end this section just taking a look at some of our antibiotic resistant strains. So these include things like Staph aureus, Pseudomonas aeruginosa, Mycobacterium, including Mycobacterium tuberculosis, and Plasmodium, which we will talk about at the end of the week, is actually not a bacterium, but this is a parasitic infection that causes the disease of malaria. And so we'll focus on this towards the end of the week. So I think I will stop here for this section.